Hello, everyone. Welcome to today's CCMB department seminar. So today we are really fortunate to have Dr. Wilson to give a talk. So Dr. Wilson is an assistant professor in psychiatry at the University of Michigan. He actually grew up in Ann Arbor and then obtained his BA from Cornell University and his MD PhD degrees from Columbia. During his PhD, he used two-photon microscopy to study the behavior of neurons in local cortical microcircuits. During his doctoral work, he also participated in the technical development of multi-beam two-photon imaging technology. Upon graduation from medical school, Dr. Wilson pursued a residency in psychiatry at Weill Cornell Medical College, as well as postdoc work at NYU. He received the National Institute for Mental Health's Outstanding Resident Award, the American Psychiatric Association's Lilly's Research Fellowship, and the Leon Levy Neuroscience Fellowship. She did a fellowship with Dr. Georgi Bazaki at NYU to record ongoing activity in natural behaving and sleeping animal, wherein he showed that sleep reorganized neuron firing architecture in the neural cortex in previous unknown ways. He's now combining his electrical recordings with behavioral tools to deepen his understanding of both use and regulation of the cortical brain circuits. So without further ado, let's welcome Dr. Wilson to deliver the talk titled Recording from the Neural Circuits of the Cortex Across Varying Brain States. Welcome, thank you.
Thanks so much, Dr. Liu. I really appreciate being invited to speak. I might have shortened that bio if I knew you'd actually read it, so I apologize. But thank you so much, it was very nice. And I guess I'll just jump in. I will just explain briefly at the beginning. Well, actually, first, I think I was asked to talk about how we should do questions. I have not tried Zoom lectures with sort of questions in the middle. We could try it. There are probably three natural stopping points, two in the middle and one and then at the end that I could let you guys know about, and then there could be questions at those points. That's probably a pretty efficient way to go. At that point, by the way, right now, can you see the PowerPoint or did it stop? It's okay. So when I'm looking at Zoom, it's not working. Okay. So I'm gonna let people just ask me questions verbally then instead of using the chat, because when I scroll over on my Apple, you guys can't see the slides anymore. So I'll tell you when there's some stopping points. And we can do questions, and then I also just wanted to explain a little bit that I'm going to be talking about work that's a mix of what I did in my postdoc and what I've done here since coming here in 2017. Really work, obviously, that students and postdocs have done and not me. I thought that it would be actually a bit more coherent if I did it that way instead of just focusing on what I've done since coming here to try to make a little bit of a more linear story for you guys. So as you can see from the title, I'm interested, I do recordings, I do electrical recordings in the brain, and I'm interested in kind of as the brain changes states, and I'll tell you what I mean by that, how do neural networks change? So the brain, so if we zoom in, on this, here's an MRI picture of the brain. If we zoom in really small, right, really high power, we can see in a part of the brain, we have thousands of neurons, right? And these neurons can be organized in different ways. For example,
Brendon, we can't see the slides.
Oh, you can't see those at all?
No.
Oh no, okay, that's bad. Let me go back and share the screen. Thanks for speaking up. Let's cancel and restart the screen share. Okay, now can you see PowerPoint?
Yes.
Okay, good, yay. Let's try to just, okay, and full screen?
Yes.
Good, all right, progress. All right, here's the brain. We zoom in, there's neurons, and the neurons can have various kinds of structures that we're not gonna concentrate on too much, certainly at the beginning of the lecture, but these are the networks that inspired artificial neural networks, these cortical networks that are found in this outer part of the brain, the cortex, have different kinds of connectivities. So this is a 100-year-old drawing from under a microscope of just what you see if you just look for cell bodies of the neurons. And then over the last 100 years, people have done lots of really good work to map out the connections between different types of neurons. And so these lines kind of show that different types of neurons tend to have different connections with other types of neurons. And so we think these networks really are functioning almost like, I mean, different neuroscientists will argue this point to some extent, but kind of like computers, right? Like little computing units. And the question is kind of as the brain, so one of the questions we've been looking at is as the brain goes through the day, you're awake, you're asleep, you're paying attention, you're stopping attention, all sorts of things are happening. And how are these networks functioning differently under those different states? What's the purpose of these brain states, these attentional states, these sleep states, REM sleep? How does that actually affect these neural networks? So let's sort of take you through our work looking at that. So one of the things that if you just put an electrode on the scalp of a person and do an EEG recording, or if you put an electrode inside of the brain also, you see these oscillations. And the only real takeaway message from this slide is to show that there are oscillations across different species, and there are different kinds of oscillations. Actually, the way to see that there are different oscillations is that these time scales are different. And so we have these kind of oscillations that are happening in the voltage that you can just record out of a brain, kind of like an EKG from the heart. That's all you need to know for this slide, right? That there's these oscillations. We don't know exactly what they mean, but we're working on them. They seem very interesting. They're organized. And so kind of the next finding is that these oscillations change depending on what a person or an animal is doing, depending on their behavioral state. And this is the first kind of look at states. So for example, here's a rat that's, well, if you know rats, you'd know that it's sniffing and it's flicking its whiskers around. It's trying to take in information. It's looking around. It's exploring what's outside of this box that it's in. And here's the same rat, maybe a couple minutes later, sitting and just kind of resting, or it might even be scratching it, itself or cleaning something kind of absent-mindedly. I mean, these are very different states. This is an exploratory state. It could be walking or looking or moving, gathering information. And here's another rest state where the brain's doing something else. And in these two different states, taking in information versus maybe consolidating old and reprocessing old information, we have different kinds of oscillations. So, and you don't need to know these names, but they've been given names. Here's an oscillation though that's kind of has this relatively reasonably regular rhythm. This is biology, so we can't get too perfect. And then we go from this when the animal's exploring and taking in information to when it stops, we have this other kind of brain rhythm where we have these irregular, bigger things called sharp waves, right? So these behavioral states correlate with different brain oscillatory states. And we can even look one step deeper into an even bigger change in brain state, which is sleep. Sleep is probably the sleep-wake dichotomy of brain states is probably the biggest, the most separated pair of brain states we could probably say, right? So you're awake, you're active, you're moving versus you're asleep and totally not engaged in the world. And those are also characterized by very different oscillations. When you're awake, you have these tiny little low amplitude oscillations. They're not big waves, they're small, and they happen very frequently. So their frequency is high. Whereas when you're in the deepest levels of sleep, you have these big delta waves, these big oscillations that only have, the cycle time is much longer. So to go from this peak to that peak is a lot longer than from one of these tiny peaks to another one of these tiny peaks. So we have these differences in the frequency, kind of how often waves happen, and in the size of the waves. And so we can quantify that to look at sleep, to automatically score sleep. And that's one of our first tools that we're going to use is we're going to record an animal for hours, record it from its brain for hours. And I'm going to explain how this plot works. But basically what this is showing is in red, the animal's asleep. And when there's not these big red blocks, the animal's awake. And what this is it's called a spectrogram, and it's basically representing this oscillatory information graphically. And what it says is there are different frequencies of oscillation, just like these high frequency and low frequency. And the color tells us the power. So basically what this is showing is at this time, the red is showing us that there's a lot of power in these low frequencies down here, around whatever, 1 to 32 hertz kind of area, meaning that the animal is in this sleep state. And the animal wakes up, or maybe goes into REM or something, has a little bit of a different state change, and these big delta waves are gone, right? So we can automatically score, is the animal asleep or not, pretty easily using some basic classifiers. I'll come back to this at some point. But suffice it to say, we have these two very different brain states. It's very easy to represent graphically how different they are. And we were curious to understand more about it, right? So I'm a psychiatrist. I work in the department of psychiatry. I see some patients. And we know as clinicians that sleep is crucial for health. It's both in terms of pathology and in terms of just optimal functioning when you're not in a pathological state. So here's some poor lady's to-do list that I got off the internet, right? So there's a lot that we do every day. Got to do all this stuff, right? Sort out your purse in Amsterdam and whatever. We are busy. And then you go to sleep and your brain has to help you get ready for the next day. By the time you're going to sleep, you're tired, you're not as productive as you were in the morning. There are other fluctuations in the day that we know about, but if someone gets sleep deprived, a lot of things change. And that tells us that sleep matters. So for example, if you don't sleep, you'll perform, there's all sorts of actual real tests that have been done, published studies, but we all know too, subjectively, if you don't get sleep, you're not going to think as clearly. You might get your moods might you might get irritable, or you might laugh extra easily, things like that. Things change in our mental function, and they can lead to bigger problems that actually sort of reach the level of clinical problematicness, right? So manic episodes in bipolar disorder, for example, you can get psychotic, which means you lose touch with reality, you start hallucinating or being delusional, having ideas that things aren't happening. I've seen a couple, probably 3 to 4 patients where sleep deprivation alone has caused them to have paranoid delusions that people are coming after them, that really they've been followed for the last couple of days. They don't know who it is, just from sleep disorder, and just sleep disruption, and you get them, it's rare, but it can happen, right? And you get them to sleep enough, maybe you give them some Ambien or something, and it kind of goes away. And sleep disruption is also seen in a lot of major disorders, both neurologic and psychiatric. So depression, anxiety, PTSD, bipolar, and seizures can happen more, which are known to be very brain network based, even on a sort of micro scale, if there's sleep deprivation and you have epilepsy. So sleep's important, but we don't really know much of what it does, right? So one of the major theories about how sleep maintains proper function is this synaptic homeostasis hypothesis. So this maintaining a proper function, that's obviously a version of homeostasis. We don't know how the precise details of that homeostasis, but we know it's keeping something working properly, something in balance if you sleep every day. And one of the major theories about how the mechanism of how that sleep is helping is this hypothesis that with wake versus sleep, we have very different changes that happen in the synapses that connect the neurons. So for example, here at the beginning of the day, we have these 2 synapses. That's what these 2 little graphics are representing. And they have given a synaptic weight of 100 each. By the end of the day, we might have learned something, and one of our synapses might have grown larger so that we could, understand that every time that dog barks, means something scary is coming, right? Whatever. So you've built a synapse connecting 2 pieces of information in your brain, and you might have even put in a new synapse, and this other one hasn't changed, right? So we learn things, change our synaptic weights. We know that as neuroscientists. And then when you sleep, these synapses downscale. So this is the theory, right? These synapses become weaker to bring their total strength back to 200. So at the beginning of the day, they started at 100 plus 100 equals 200. At the end of the night, at the beginning of the next day, they're back to a total of 200 so that the system doesn't get out of balance, doesn't get epileptic, overly excitable. Although these people are very smart and they include the relative weighting that was acquired after learning, right? So you don't ruin the learning, you just downscale back. And actually now, since this theoretical work was done, this has been shown to be the case with electron microscopy with synapses. With neurons, however, which are obviously what's being connected by the synapses, people had assumed that there'd be a similar story where when you sleep, if you go from early sleep to late sleep, neurons become less active as their synapses become smaller, as the synapses become downscaled. So here's some recordings that was done in the 2000s where early sleep to late sleep, the firing rate, I haven't even explained what spikes are, right? But neurons fire these spikes, these action potentials to send information to each other through their synapses. So these are basic markers of neural activity, of neurons doing things and talking to each other and sharing information. And that decreases from the beginning of sleep to the end. And so what we did was we took that idea, we said, okay, let's see if we really see that in a different part of the brain. That data that I just showed you, this one here was acquired in one part of the cortex, and we were curious to see if this was a general phenomenon. And we ended up finding something a bit different. I'll sort of preview that. We actually confirmed their results, but then we add some dimensionality to it. So to do this, we use these probes, since this is a new slide deck for me. Okay, so we use these probes where we can implant them into the brain. So this purple thing is the brain of a rat, and these little yellow dots are neurons that are stained. And what we can do is we can put in these things that are actually called, the world over, they're called Michigan probes. They're silicon probes is what they're called. They're designed here at the School of Engineering and are actually made by a company out by the Ann Arbor Airport called NeuroNexus. And this is the most popular company in the world for these things, these silicon probes. So we use silicon probes when I was in New York, and we put them into the brain amongst the neurons, and they have these, they might have 8 different, these things called shanks. And at the end of each shank are 8 electrodes, that 8 recording sites. So we have a total of 64 recording sites, 8 on each shank and 8 shanks. And what we can get out of it is these voltage traces. So we have multiple voltage traces here. I believe this is 64. And so each of these lines is a trace of voltage that comes from one of these electrodes. And what we have is a mixture of these oscillations that you can see here that are kind of at a relatively slower time scale, and then these very fast time scale spikes. These are the spikes that I was talking about, the neurons sending to each other, that are signatures of neurons sending information to each other. These are action potentials. So we can record action potentials, and we can record action potentials from many neurons, because as you can see, these different recording sites are in slightly different location, and these gold balls are different neurons. And so each electrode is next to a different set of neurons. And so therefore each data stream that we get out that's streaming over time gives us spikes from different neurons. And you can actually see that electrodes that are near each other might see the same spikes. And we actually can use that we can use that in some clustering and triangulation methods to localize specific neurons. But we can record multiple neurons at once this way. We get really rich data sets over hours. Okay, so we recorded from the prefrontal cortex, because again, I think this is really interesting as a psychiatrist, a place that seems to control a lot of our moods and our decision making. So we put our electrodes, this is a cross section of a rat brain here, and here, right here is where we put our electrodes, our silicon probes, and we actually put them in a variety of places around the frontal area of the brain. And I guess I can't show you, but if you went into the screen, you'd be more frontal, and if you come back out of the screen, you'd be more posterior in the brain. So all of this is frontal, it's just a cross section of the front. And then we can record neurons with different types with different waveforms, and we ended up concentrating on the most common type of neuron in the cortex, which is the excitatory type neurons that have these wider spikes. So these are the actual shapes of the actual action potential spikes themselves. And we concentrate on these wider types of waveforms that come from the 80% of neurons that compose most of the cortex, the pyramidal cells or excitatory cells. So if we record from these excitatory neurons, I apologize, I don't have preview mode, and this is a little bit of a new arrangement of slide deck for me. So I'm just going to kind of jump and hopefully I'm setting up the next slide properly because I don't have presenter view, which is what I had tried to do the first time. So we're looking at these excitatory cells. We think that these are the cells that carry the information for the most part. They're inhibitory cells that are the other kinds of cells. And we think that, to put it simplistically, that maybe they just control the excitatory cells. That may be not quite true, but the excitatory cells are the ones that seem to know whether you're seeing red or blue. They know whether you're seeing a line that's this orientation or that orientation. So we thought we would start with those neurons. And so what we first did was to try to replicate some of the findings I showed you earlier. We wanted to just look at from before sleep to after sleep, or from the first third of sleep to the later part of sleep, first third to the last third. Do we see a drop in spike rates? And yeah, but it's really messy. So this is either just the raw data or the subtraction data. So we see that, yeah, there's a drop in spike rates, but it is a giant mess, right? Like there's this error bar is way bigger than the actual signal. So, but this is a statistically significant drop in firing rate from before sleep to after sleep, or from early sleep to late sleep. So we confirm what was found by those previous researchers. But this error bar got us really interested. What is going on? Actually, I should say concerned, and I thought I was doing something very wrong. But we started dissecting it, right? We started dissecting the error bar. And so what we used is this idea of the fact that the error bar might relate to the fact that neurons are not all the same. We hope they're not, and we know they're not, right? If they were all the same, they'd all be representing the same information, and they'd all be redundant with each other. We, in fact, know that they're actually quite different from each other. And so treating them as one type of thing, one uniform group, might not be the right approach. And so as an anchor to try to understand more about the diversity of neurons and whether maybe that plays into that giant error bar, we took advantage of just a really basic fact. So if you just forget information, so there's a lot of neuroscientists, and actually later in this talk, I'll talk more about encoding specific pieces of information by specific spikes. So for example, a spike that again discriminates colors, for example. But if you throw that away and you just back up, take a wide view of your data, and you just say, let me just see how much one cell fires versus another cell. So we could here look at this data, and there's these little tick marks, right? And each tick mark is a spike, and the x-axis is time, and the y-axis is different neurons. And we can see different neurons are firing over time. And some neurons fire less, and some neurons fire more. And if we plot a distribution of how those neurons fire, of how neurons in our recordings fire, we see something like this. We see basically a log normal distribution, or maybe a gamma distribution for those of you who are really into these things. So we see that most neurons tend to fire at about 1 hertz. There's a few neurons that fire a whole lot. There's a lot of neurons that fire a little and a few neurons that fire a lot. So but regardless, there's a distribution. There's a distribution of basic firing rates and activity levels. And as you remember, we were just talking about firing rates and how neurons are active or not active before versus after sleep. So the fact that neurons are really diverse in their basic firing rate made us think that maybe we should look at this more carefully. So if you log transform this histogram, it looks somewhat more normal. I'm actually going to, I'll briefly mention that these log normal distributions, or these very widely, very skewed distributions, exist in lots of parts of the brain, not just in firing rates, but in the size of, so this is a firing rate. This is from a different part of the brain and inspired some of our ideas. This is from the hippocampus. But also if you look at the sizes of the synaptic inputs that neurons give to each other, they're log normally distributed. The sizes of the actual physical synapses are log normally distributed, sizes of the axon distributed. And that's really interesting. It actually might be good for something. We think it actually might be good for encoding a variety of types of information. I don't know how many, I don't know that much psychology, but if some of you know more than me, you might know about the Weber-Fechner law, which is that there's actually a log relationship between perception of stimuli of different amplitudes, right? So you may see light that differs by a factor of 10. Let's say you have some light of 1 lux versus 10 lux. Those two might look about as different to you as 10 lux versus 100 lux, right? Not 10 versus 20. So the relationships in terms of how we perceive things, including some numbers, is there some numerosity studies and people's ability to discriminate numbers. In untrained people, they see that basically in Western people who've been trained to count by ones, you can discriminate 5 versus 6 just as easily as you can discriminate 1 versus 2. But in someone who hasn't been trained to count in our linear system, they'll basically use a logarithmic version of perception where they can distinguish percent changes basically. And so these logarithmic distributions of elements of the brain itself may be used to help us encode a whole range of different kinds of stimuli. So anyway, so we have this distributed, I just think that's interesting and I like saying it, so right. So we have this log normally distributed or very skewed distribution of neuronal firing rates. And what we ended up finding is that these firing rates end up sticking as pretty good labels for a given cell. A neuron's going to keep its firing rate over the course of a recording. So we can kind of say, this is a high firing rate neuron, this is a low firing rate neuron. One of the tests we used to decide whether these are good and relatively permanent labels was just to look at the firing rates of a neuron. So each of these dots is one neuron and how much it fired in when the animal was awake versus when the animal was asleep. And there's a tendency that if you're going to be a high firing rate neuron in wake, you're going to be a high firing rate neuron in non-REM sleep, for example. This, by the way, is actually quite a nice way to discriminate these neurons that we have previously. There's 80% of neurons that we previously haven't had too many ways of discriminating on a functional level. So what we did is we took our data and we took our neurons that had different firing rates and categorized them just into bins, at least as a first example. So we had some neurons that were the highest firing here and some neurons that were the lowest firing and different bins in between. And so what we did is we just looked at the firing rates over the course of wake and sleep. I'm just gonna move this zoom box. Over wake and sleep in long-term recordings. So in black are times when the animal's awake, and in blue is the times when animals are in the kind of majority of sleep, which is non-REM sleep, and in red is the relatively exceptional REM sleep. And what we could basically see is that when the animal's awake, the neuron's firing a lot in these high firing rate neurons. When the animal goes to sleep, those neurons slow down their firing rates, just like what was shown in the previous paper. But on the other hand, when we look at the lower firing rate neurons, we see the opposite relationship. We see that when the animal's awake, these neurons actually fire less. And when the animal goes to sleep, they speed up, they fire more. So we have these very opposing relationships between these kinds of neurons. And this seems to be where those giant error bars came from. Let's see, yes. So if we take, if we take this phenomenon of the high firing rate neurons firing more here in wake, and then slowing down versus low firing rate neurons speeding up during sleep, and we just average a whole bunch of these wake to sleep transitions, we see this over the course of our whole population. So we see, again, high firing rate neurons slow down during sleep and low firing rate neurons speed up during sleep. And if we just take the whole population, and we see that the whole population on average does drop its firing rate over sleep. So this is the beginning of sleep here, and this is the end of sleep here. So we just took a sort of percent of the way through sleep or a proportion of the way through sleep. And we see that overall, we do see a lowering of firing rates. And that's because, just by logic, these high firing rate neurons have more spikes than the low firing rate neurons. And so their effects outweigh these low firing rate cells. So these high firing rate neurons, by the fact that they have more spikes, will mean that overall the population has fewer spikes over the course of sleep. But that there's another dimension to this is quite interesting. And this was our main finding. We got rid of our bins and instead just did just a correlation between how sort of firing rate versus firing rate change, basically. Well, this is firing rate at the beginning of sleep versus firing rate at the end of sleep. And we see that there's a statistically significant tilt. It's not a huge change because these labels are pretty permanent. So this high firing rate neuron is not going to suddenly drop all the way down, right? You notice that these lines don't cross. But just that when the brain's regulating itself using sleep to keep things balanced, to keep our brains working properly, it's not just lowering the firing rates, it's homogenizing them. We did a different kind of quantification to show the same thing, that even from the wake before sleep starts to the wake after, we have this same net change. And we actually also tested for a reduction in the width of the distribution, which we also see. So we wanted to actually go into the details of sleep. Let me just check the time and see whether we want to go into this depth. Or 32. Let's not go super deep on this. We looked at the different parts of sleep. So there's REM, there's non-REM. Every time you go to sleep, you start with non-REM and you go to REM. And this is a classic kind of illustration you might have seen in a textbook. And we have automated means to classify REM and non-REM using these spectral, right? These frequencies of oscillations that are present. And what we find is that REM and non-REM are very different from each other in wake, right? So wake widens out the distribution of firing rates of neurons. Non-REM squeezes it down the way we saw that most of sleep does, and REM does something different. REM actually kind of just temporarily bumps up the firing rates and then they come back down again. And there was one other part of sleep that we ended up investigating, which is these little things called microarousals that it's not clear whether they're healthy or unhealthy, but we actually found that they occur very regularly and that they occur at something called an infraslow oscillation rhythm, which is about 1 minute. So they actually seem to be a regular organized part of sleep. They're not only present the way sometimes clinicians see them, like in sleep apnea cases where you can't breathe. They actually seem to be a regular part of healthy sleep as well. And so we found that these different parts, I'll go just zoom to this slide because it's a little simpler. These different parts of sleep play different roles in regulating this overall homogenization of firing rates that happens during sleep. So we have this black line showing where the way spike rates are during wake. And then by the end of sleep, we have this squeezing together of this distribution on this gray line. And what we actually find is that non-REM most squeezes the top firing rate neurons in, and these microarousals actually have a net effect of squeezing in these lowest firing rate neurons in from the left. And so basically the non-REM and the microarousals work together to have this net homogenization effect of sleep. So these different parts of sleep seem to be doing different things. And REM is always a mystery and has continued to be a mystery. I'm not going to talk about it today, but we have some new findings in the lab kind of starting to dig into some of the microstructure of REM and some more details about how it's actually built. But as usual, it's doing its own thing. It's like a little bit off the grid, but maybe it's part of this net downscaling. Maybe it's a little bit different. And so we would just modify this prediction that comes from the synaptic findings. They would have predicted that there's just a general downscaling of activity over the course of sleep. We say, no, it's not a downscaling of everybody. It's just a downscaling of the most active, and it's an upscaling of the least active. So this is, again, like I said at the beginning, kind of a new dimension. So I could actually draw this a little bit differently. There's actually a bit of a net downscaling, but there's an additional homogenization. I'm not going to talk about mechanism with this awesome student who's actually defending tomorrow, if anyone wants to see his defense from the Buzaki lab at NYU. But we have some ideas about the mechanism for this squeezing together, which I'm not going to detail. So we have this finding that brain states matter. And I think I'll stop there if there are any questions before I go on to the next part, which is a little bit more about the functioning brain and the states of within wake, like attention, motivation. I know there may or may not be questions, but certainly anyone who has them, go ahead and ask away. Otherwise, I can go ahead. I'm going to check the chat or Q&A.
So there is a there's a question from the chat
Yeah.
from Gil Oman.
Okay.
So Gil, are you still around? Do you want to ask
I see.
yourself?
Oh, I see it now.
Oh, okay.
So Gil asked, you use deep learning or machine learning to segment and distinguish wave patterns in these different brain states. So that's a great question. We actually assumed we would have to. So I'm going to just go back to, I think, what he's talking about. So to segment, this versus that. And we thought we just didn't have to. So we were able to do a much simpler, we just did PCA as our first way of doing things. I don't have a full plot, but basically we can do PCA to pull out this component that kind of shows, hey, that I'm asleep. And then we basically find that there's a bimodal distribution of the power of the projection onto the principal component that sort of most strongly comes out of these times. So basically you can take these times and see how you can idealize it with a PCA and then do a vector projection of every single second of time onto this idealized sleep principal component and you find that basically sometimes there's a really strong projection onto that and sometimes there's a very weak projection onto that state. So we thought we'd have to, but we didn't, and so we didn't. we didn't have to, so we didn't do it, and we just kind of moved on with our experiments. Pragmatic. I think that's it, so I can go on. So the next part is, is more related to brain function. And I think I had a tiny motivating slide here. So we had spiking, but we kind of, as I said, pull them away, the spikes away from, what we think they're used for, what we think they're doing. We think that they, that there were some Nobel Prize winning experiments in the 50s and 60s from Hubel and Wiesel that showed that, when you show different kinds of stimuli to a cat, for example, visually, and you're recording from the visual part of its brain, the neurons there are very specific to they'll fire action potentials very specifically with some stimuli and very much less with other stimuli. And people use the word neural coding kind of came from this, right? That this neuron is encoding when there's a line of this orientation and the other neuron fires and therefore encodes when there's a line of this orientation. And the population overall of those neurons encodes orientation. And again, this is kind of a lot of neural network models came out of this kind of work from Hubel and Wiesel that, if we plug in certain kinds of stimulus specificity to certain elements of this artificial neural network, and we have them talk to each other and share information and then randomize things, maybe we can get, different stuff to happen. And now the field of machine learning and neural network based learning is, taken off on its own, and it sometimes has nothing to do with neuroscience and sometimes maybe even can teach neuroscience. And there's a kind of interesting, anyway, so these spikes, we think they're important. And in the first work, we were just kind of taking this bird's eye view of what does sleep do to this really basic global phenomenon of spiking overall, like of talking, like what's the volume of the conversation basically. And so what I'm going to talk about now is work done by Alessio Bueno in my lab here at Michigan. And we're not going to get to any final answers, but we're going to start, I'm going to talk to you about the beginnings of some experiments to start to look at how these firing rates are changed as an animal goes through different waking states. I'll tell you what I mean. So first of all, I'm just going to mention in the previous experiment, we were recording up here in the frontal cortex, which has more to do again with these sort of mood and psychiatric probably things, right? So regulation, decision-making. And in this next set of experiments, we intentionally move away from the prefrontal cortex to this much better studied area called vagal cortex. Or it's the part of the brain that has to do with somatosensation or feeling of touch. That's one of the five senses. And in this case, these are the neurons dedicated not just to any touch, but to whisker stimulation. And it turns out in rodents, they use their whiskers kind of like we use our hands, or maybe even more so, right? They will just kind of, because it's on their faces and they don't have great vision. So they'll kind of just stick their snout, they'll be sniffing and doing this thing called whisking, which is like active searching with their whiskers for stuff, and feeling what's right in front of them and understanding textures from it. They can do incredible encoding with their whiskers. It's kind of like sweeping your finger over something. If you just put your finger on something, you may feel it for a second and then the feeling goes away. But if you keep sweeping it and whisking, you can keep feeling the texture. So there's this very overdeveloped part of the rodent brain called this barrel cortex. And each of these, it's called barrel cortex because there are these structures, these columns of cells, and each column or each barrel is dedicated to one whisker. So it's super organized, and neuroscientists have done tons of work. And so, my PhD work, we studied the cortex, and this part of the cortex in particular, because it has this really nice layered organization. All the parts of the brain have, all the parts of the cortex have a layered organization, but it's best understood here. And I decided that if I want to really understand even the prefrontal cortex, I need to understand the layers. I'll show you what I mean, right? So here's that barrel cortex, and these stars are some of the barrels you can see even without any staining. With staining, they're beautiful, but even without staining, you can kind of see them. And as you might have seen from the old drawings that I showed you from 100 years ago, there's kind of this structure to the neurons. This is the surface of the brain up here. This is a deeper part of the brain down here. And this is white matter, right? And there have been given these, there are layers that have been given names 1 through 6, because with different kinds of stains, you can see different microstructures within the anatomy of the brain, of the cortical cortex across these layers. And lots of work over years has figured out that there are synaptic connections between neurons in these layers, kind of like in that diagram I showed you. So we have this over decades, it's been built up this nice diagram that, hey, when a sensory input comes in, let's say from the whiskers or from the eyes, it comes first from the whisker to the thalamus, and then from the thalamus up to layer 4 of the cortex. Just, we don't have, there's nothing to know. It just goes to layer 4. Like, that doesn't have any special connotations to neuroscientists. It is. So it goes to layer 4, and then they go up to layer 2, 3 for some reason, and they go to layer 6 for some reason. So if you just map the synapses, this is where the predominant synaptic projections are. Again, we know, though, that the brain is oscillating in these different ways. And we wonder how, as these oscillation changes, information flow and spiking might change across those layers. And it's actually, there's already been some findings from one of my favorite labs, this guy, Ken Harris. He showed that under different brain states, if we look across the layers, and so what he did is he put some electrodes across the layers, there are different patterns of information flow across those layers. And I'll come back to some of the implications of that in a second. But so, for example, this is an auditory cortex, when he gives a stimulus, he sees, or like it's the same as flicking a whisker, he sees that activity starts in these 2 places, layer 4 and layer 6. Whereas when there's a spontaneous event called an upstate, the activity starts more just down here in layers 5 and 6. So despite this sort of very strongly evidence-based structure going from layer 4 to layer 2, 3 to layers 5 and 6 in terms of where the synapses go, actually information spiking can kind of spread from one place to another in a much more dynamic way that hasn't been studied in very much detail yet. And that's probably because these anatomical diagrams are really just the strongest synapses, but they're not much stronger than the weaker synapses, which go everywhere. And a lot of computer modelers actually model cortical circuits. And again, artificial neural networks are also modeled as basically all-to-all connectivity circuits. So any network, any neuron anywhere in this network can actually have a pretty decent chance of being connected to any other neuron anywhere. And just that there are these slightly stronger tendencies of connections from the thalamus to 4 to 2, 3 to 5 and 6. But these aren't the only connections. And so we're very interested in understanding, again, if we put one of our silicon probes across all the layers at once, can we see a variety of different kinds of activity across these layers to kind of contradict this idea that there's just one static network, and to see if there's a really dynamic network that's used in different ways depending on the situation. And I won't be able to tell you the end of that story because we're not done, but I'll start with it. You can skip. So in order to study this, what we're doing is we're using mice, and we're using this commonly used technique of what's called head fixation that came from actually 2-photon imaging people and other people. You put them in here, and what you do is you train them to be pretty happy with actually being in here because you give them rewards over and over. And so they go from initially being somewhat distressed to later not seeming to resist at all going in there because they always get water and treats and things when they're in here. And what we did is we mixed that with my previous background, which is recordings of an animal as they freely move around. So there's obviously not a rat, by the way, it's a mouse. But we chose mice for some specific mechanical reasons. But so we have this mixed model of the way some cortical physiologists usually record in this head fixed fashion, and the way the hippocampal people usually measure, and the way I was trained by this hippocampal lab, freely moving natural sleep. So we want to mix this nice head fixed method where we can teach animals and give them really precise stimuli into their whiskers with natural waking and sleeping. So we have kind of this mix, and we can do that in the same recording, this mixed recording method. We do lots of monitoring, which I don't have time to tell you because I've been talking longer than I should have. But we record things like how their eye is, pupil is, right? Are they excited? Are they getting drowsy? We can watch their whiskers as they flicker back and forth to do that whisking to gather information. We actually stimulate their whiskers. And what we're doing is we're giving them different kinds of stimuli to learn so that they can learn a task. So the task is, it's called a go, no-go task. They can either choose to lick for water or not. And these are animals that get water on this head fixed rig, head fixed recording system. And they're restricted for some of the other hours of the day so that they get nice and thirsty before this, right? And we say, if we take your whisker and flick it backwards while we're recording from your whisker system, that means you're allowed to lick for water. If we flick it forwards, you're not supposed to lick, you're supposed to wait and not lick. And if you do lick, you have to wait longer for water the next time. There's a little 10-second timeout. So it's a task, and they eventually learn to do it very well. So over the course of 14 days, we were able to train an animal from doing relatively randomly at this 0.5 level of performance. So they randomly choose to getting up to 0.8 or 0.9. What we sort of percent correct. What we find is that they're really, as you can already see in this data, the behavioral data, there are these huge, this is a day 10, day 11, day 12. There are these huge oscillations or fluctuations within a day. In the middle of each day, they're performing really well at these high rates. And then at the beginning and the end of the day, they're not. And we can show an example day here, where at the beginning, they're performing poorly. And at the end, they're performing poorly. And in the middle, they're performing well. Whereas on day 1 of training, they're just performing at basically chance the entire time. So by the end, they've learned to, for some period of time, discriminate accurately. But what's interesting is that they fail in different ways at the beginning versus the end of the training. At the beginning, they're licking too much. And they're licking when they shouldn't be licking. They're pretty much licking all the time. And at the end, they're never licking. And in the middle, they find a way to be optimal and to only lick when they are really given the right cue to do so. So we have these two different kinds of failure modes. And we think that this is an overmotivated, kind of overexcited mode. And we think that this is a disinterested mode where they've gotten enough water. And some other experiments by other labs have taken this disinterested mode and done some optogenetic stimulation of the hypothalamus, which part of which controls water and thirst, and found pretty good evidence that this really is a satiated state where they're just not thirsty anymore. And so what we're starting to do, that I won't get to tell you the end of because it's not done, is to look over the course of these multiple days. So now we have days on the y-axis. And over the course of 500 trials each day, we see that the probability of correct responding goes up in the middle of the session as the animal is trained. So at the beginning, the animal is pretty bad. And by the end, in the middle of every day, they're pretty good. And then what we can do is we're simultaneously recording from that barrel cortex and from across all the layers of the barrel cortex. And so we can either measure things that are physiological and kind of like pupil diameter, wheel running on their little running wheel that they have under them, like rates, eyelid separation, or we can measure things actually in the brain. And here's probably the most interesting one, spiking metrics. What we find interestingly is that, so let's take these train sessions where they supposedly know what they're doing, and we can look at the brain state evolution. So again, here's our correctness. So at the beginning, they're not correct. In the middle, they are, and at the end, they're not. At the beginning, we find that this is essentially a measure of how much neurons are specifically firing to stimuli. And at the beginning, the neurons are not firing in a stimulus specific manner. They're not firing differently to the forward versus backwards stimulation of the whisker. They're not discriminating. Even the neurons aren't. In the middle, the neurons start discriminating very well, forward versus backward deflections, go versus no-go stimulations. And interestingly, at the end, when they're performing poorly, the neurons are discriminating. And so the story looks like it's animal and brain really respond to states differently. So this is some kind of over-motivated state where, I don't know, the neurons are not even encoding things right. In the middle, they're encoding and performing properly. And at the end, the neurons are encoding, but probably the animal's just not motivated anymore. And so it doesn't finalize proper licking. And finally, we wanted to look across layers. And what we find is that, so this is layers here on the vertical axis. And what we can see if we look across training, or if we look across brain states, so the early part of the session where they're hyper licking and not accurate, mid session where they're accurately performing, and late session where they're under licking and not accurate, we see that these are neurons and how they're firing according to their layer. So vertical is layer, and each of these bars of these bar graphs are neurons. So neurons, and it sort of doesn't matter what this is. This is, in this case, this is the how much the neurons are specifically modulated by the stimulus, just like I showed in the last plot. But we've done this with other measures too. We find that neurons in certain layers are kind of modulated together. You can see this spatial cluster, right? And then there's a bunch that get modulated oppositely in late session versus early session. So we have these spatial clusters where the cortical layers look like they're being modulated differently as the animal's learning, and as the animal, or really in this case, as the animal's going through, as a trained animal is going through its hypermotivated to optimally motivated to undermotivated states. And we are, definitely right in the middle of this and haven't dug more in, but that's kind of where we're going. And I am out of time. I was asked to give about a 50-minute talk, but I will just briefly mention some other efforts that we're making, probably in like a 1-minute time, which is that, again, I am a psychiatrist, and so I want to take some of this neuroscience and move it towards something that can start getting us towards understanding psychiatric disease a little bit better. The problems are many, but we've taken advantage of some interesting findings in psychiatry recently with this drug ketamine. Turns out within about couple hours, it can make people feel a lot less depressed, and even with a single injection, it can keep you feeling less depressed for a few days. And so we decided to do some recordings in mice that we give ketamine to, because it's behavior. Other people have done behavioral work to show that ketamine shows behavioral effects in mice similar to they do in humans that last over days with a single injection. And we find is that networks change in lasting ways. So I went back to the prefrontal cortex and found that actually the same thing I showed you during sleep, the width of the firing rate distribution actually goes up 24 hours after ketamine compared to where it was at on a baseline circadian matched period before ketamine. And the last thing we're doing is we have these living environments that we're building that we just call boxes, but behavioral boxes, something like that. Digital homecage, sometimes we call them, where we can measure, we think about 50 things at once as we record from the brain. And so we can try to integrate biomarkers of stress states that are much longer term than these little micro states I've been telling you about, and how those might correlate with spiking in the prefrontal cortex and in other parts of the brain network. We might even be able to link up some of those physiological things that we can measure over, let's say, weeks in a mouse with things that we can measure over weeks in a person that a lot of people here do, including, I think, Danny Forger, who I saw, and Srijan Sen and some other people, looking at, you take a person and you stress them and see how their sleep changes, see how their activity levels change, how their heart rate variability changes. We might be able to start doing the same thing in rats to build bridges between the neuroscience we can do in rats, the biomarkers we can have in rats or mice, and the biomarkers we can have in people to try to bridge all the way across from brain mechanisms human states of stress by stressing mice. That's, and I'll skip all this, but I'll stop. Thanks to, I should have kept part of my old thank you slide, which has my mentors on it, which Yuri Buzaki, Rafa Yuste, Jack Barkas, and Francis Lee at Cornell, who helped me do a lot of the postdoctoral work. And then my lab now has just a bunch of really awesome people. It's been really fun. And so like you say, as you can see, we're in the middle of kind of really forging our own ground in the lab, but it's going well. And I can stop there. Yes, you can go to my website. If you want, I'll stop. I do see some chat questions that I can take from John Myers. Is there any evidence of sleep state related phase coding, or spike field coherence between the action potential spike trains and the dominant LFP frequencies like delta and theta? I will try to explain some of this. So perhaps understanding this relationship could elucidate the role of LFP in population level spiking during sleep states. Okay, so I was trying to give a talk for non-neuroscientists intentionally. I am happy to actually delve into this because it brings up some really interesting stuff. So the question is about how spikes and those electrical waves that I was explaining relate. And there's a ton of really cool data about that. Some of it came from the lab that I did postdoc in. So, okay, let me reread the details of the question. Spike field coherence, action potential spike trains, and the dominant LFP. So, okay, so the idea is, for sure, the answer is yes. So these oscillations, and I don't think I have slides showing this, these oscillations entrain neurons, like they make neurons fire differently. It's a slide that I skipped. They make neurons fire differently at different phases of the oscillation. So actually, how about this? Let's go to this one. So here, here's an oscillation. This isn't the most beautiful recording, but here's a little bit of an upswing, right? And here's a bit of a downswing, and here's an upswing and an upswing. And each of these upswings, actually the neurons get silent. So here's a recording from spiking of a neuron. Or here's another recording where we're not just recording from one neuron in great detail, but recording from a bunch of neurons. We can see we have these brain waves. And we see that with each wave, there's this wave of silence. And so what these dots are is spikes by different neurons. And we can see that all the neurons become silent at this time. And down here is the cumulative spike rate within a certain time bin, right? And we see when there's a wave up here, there's silence down here. And when there's a wave up here, there's silence down here. And so these waves, what they think they do is they're not just there for nothing. We think that they coordinate neurons, and we think that coordination of neuron spike timing and spiking is key to encoding complex information. In the hippocampus, there are beautiful examples where an animal's running and it has neurons that are encoding different places on a maze, let's say that it's running. And with every single cycle of the oscillation, I have a really cool slide that's not in this deck. With every single oscillation that happens 8 times per second, the animal replays where it was, where it is, and where it's going to be. And then 125 milliseconds later, right, way faster than real-time speed, it goes where it was, where it is, where it's going to be, where it was, where it is, where it's going to be, over and over and over again, every single cycle. And so these oscillations might be coordinating neurons to fire together. It may be making them repeat themselves for some reason, maybe to burn in memories. And that certainly changes with sleep, because with sleep, the main thing that changes is the size and the frequency of these oscillations. And so more is to be done, but for example, these big slow oscillations seem to be something a little bit different than those theta oscillations I just talked about when an animal's running. And I'll just quickly go to the last question because I know we're out of time. Do you think it would be possible to get these same sorts of recordings with patch clamping followed by a single RNA sequence? So actually, this picture is probably most relevant. So this is a patch clamp recording. You can record one cell in great detail. So you can record all of its spikes, and you can record its underlying membrane potential too. So it's sort of the integration of the input and outputs, the excitatory and inhibitory inputs to the neuron. And at the end of this kind of recording, you can do sequencing. So you can certainly do this. It's hard to do it with more than one cell at a time, but I think you could use it to, for example, look at, we were patched on this cell at the beginning of sleep, and we're patched, maybe you could take a bunch of cells at the beginning of sleep and a bunch of cells at the end of sleep, and you can see what's different within the cells to see if, well, sequencing. I don't know if there's a way to look at some kind of changes. I'm going to embarrass myself. I don't know if there's a way to look at some kind of regulatory changes at that time scale. I would actually think proteins might be more likely, but maybe there's regulatory changes in mRNAs, for example. I don't know if you can do that with patch-seq. That would happen on the time scale of sleep. By the way, sleep in rodents is very short. In humans, it's very long, so it's many hours, and so there's plenty of time. In rodents, I keep defaulting to thinking about like 20-minute patches of sleep because that's how that is in rodents, and that's a short time scale. So I worry that there might not be time for those kinds of changes. But in humans, you might be able to have much longer time scales. And I think, sorry, I missed one. Can you describe that mentioned optogenetics motivation study in slightly more detail? Which cell types, hypothalamic region were stimulated? Camille, okay. I would have to go back and look at the exactly, I can, if you want to type your email address to me, I can send you the paper. I'd have to go look at which parts of the hypothalamus and which kinds of neurons were there. But the first author is Alan, and it's from Science, and it's like 2018 or 19. And then there's the word thirst in it. So if you type like thirst, Science, Alan, you'll probably find it. I apologize that I don't remember the details on that one. Oh, okay, there you go. I got your email.
Great, that's a really fascinating talk. So do we have additional questions from the audience? Then let's thank Brendon again for this fascinating talk. Thank you so much for having you here today.
Absolutely, thank you for inviting me.
Thank you, and see you guys next week. Bye.
Okay, bye-bye.